Good morning, friends. I'm Dr. K.S. Dinesh Babu, Assistant Professor, Department of Botany and Microbiology, Hemavati Nandan Baguna Garhwal University, Uttarakhand. Today, I'm going to take a small lesson on plant viruses, particularly the Begomoviruses. My field of specialization is plant virology. Particularly, I am working on the Begomoviruses, particularly especially of ND11 mosaic viruses. And I'm having a research lab in HNB Garhwal University, and my lab people are working on begomoviruses. So today we are going to see some important topics about begomovirus disease symptoms and causative virus and how they get transmitted from one plant to another plant. Initially, it is better to go from the symptoms. As we are coming through many places, in many places we have seen the mosaic symptom, right? For example, in 20 plant, we have seen yellow and green color patches in the leaves. And again, everyone knows about the tobacco mosaic virus also, but this is not coming under the begomovirus. That is a kind of a different RNA virus. But today we are going to see some of the symptoms that is elicited by begomoviruses only. The same begomoviruses has the ability, has the capacity to induce these kind of mosaic symptoms. How these mosaic symptoms were elicited with the different crop species? These viruses spreads mainly through specific type of insect vectors, and once the insect vectors comes and makes a puncture in the vascular cells, this puncture serves as a kind of entry point for the viruses. Through these punctures, insect sucks the phloem cells, where the sucrose-rich phloem liquid passes through the insect stylet to the insect gut. And during this process, what happens? Virus enters with the help of this particular puncture and enters inside the phloem cells. The phloem cells are living cells and they serve as a kind of reservoir for the virus multiplication. And the virus, once after reaching these phloem cells, they start to replicate inside the phloem cell nuclei, and again access various type of cellular machineries like transcription machinery and translation machinery. Everything is available for the virus. And they start to replicate by recruiting these machineries, and viral nucleic acids increases in numbers, and new viral proteins were synthesized, and new viral particles assembles inside the phloem cells. And again, the viral nucleic acid was transported from one cell to another cells via new type of viral proteins, particularly movement proteins of viruses. And once the symptom increases, it spreads throughout the plant. It becomes visible for all the people. And this particular symptom, like a mosaic symptom, that is green, yellow, or green, white color mixed patterns in the leaves, they give a kind of clue that is easily recognizable by many people, and we can easily say that particular plant is infected with a particular virus, like that. Apart from this mosaic symptom, other symptoms also has been found. They are just leaf curling symptom. There are some plants are there. Once the virus infects, the plants cannot grow beyond certain height. So the plants remain stunted. They are very dwarf. In that case, we call this particular symptom as dwarfing symptom or stunting symptom, like that. And in many cases, leaf distortion happens. The normal shape of the leaf was modified, and again, the leaf was distorted. In that case, we call this particular symptom as leaf curling or leaf distortion symptom, like that. Here in the pictures, I have shown 4 different crop plants. These begomoviruses infects a variety of crop plants actually. Not only weed plants, it also infects weed plants, but it also infects wide variety of economically important plants. I give 4 examples here. Example number 1 is soybean. The soybean is mainly infected by a specific type of begomovirus called soybean mosaic virus, and it creates a kind of mosaic pattern in the leaf. And again, in the second case, I have shown the tomato leaf that is showing mosaic pattern and leaf curling symptom also. In this infected tomato plant, the particular begomovirus called tomato leaf curl virus is infected. And again, in the third particular plant's example, I have shown cassava plant. Here you can clearly see that the cassava leaf was disorganized, and this particular symptom is just known as leaf distorting symptom. This plant is infected by cassava mosaic virus. The fourth one, the right bottom photograph shows the symptom in the bendy plant. Ladies fingers, everyone knows about the ladies fingers. It's much nutritious and tasty vegetable. This particular plant is infected by a specific begomovirus called bendy yellow vine mosaic virus. And this virus produces a specific vein clearing symptom, and this makes a kind of a mosaic pattern in the leaf. In the second slide, I have shown the particle morphology of these particular begomoviruses. This begomoviruses contains a unique particle morphology called twinned icosahedral particles. How two different icosa particles, they join together, they stick together, and make a kind of two particles stuck in a kind of pattern. This particular structure is just known as twinned icosahedral particles. This is a common particle morphology among begomoviruses. Inside these particles, a single-stranded, covalently closed circular DNA genome is present. This DNA genome may be of 2 to 3 different kinds. Actually, in an infected plant, the major virus contains DNA A. Again, the same virus may be associated with DNA B. Again, the same virus may or may not be associated with different types of satellite molecules called alpha satellites, beta satellites, and delta satellites. These, all the DNA A, DNA B, and alpha satellites, beta satellites, the delta satellites, they encode different types of pathogenic proteins. And all the pathogenic proteins are multifunctional and they do different types of functions. And these proteins responsible for the safekeeping of viral DNA and assembly of viral particles inside the plant cells, and also for the transmission, of plant viral DNA from one cell to another cell. Here, in the right bottom, I have shown the electron micrograph of these twinned icosahedral particles isolated from the infected plants. In the right corner bottom, I have shown the insect vector Bemisia tabaci. Particularly, the begomoviruses are transmitted through this insect vector only. Once after entering inside the insect gut, the virus interacts with the host factors, particularly specific host proteins. These proteins protect the virus particle from the devastating gut juices. For example, degrading enzymes are present in the insect gut. These particular specific host proteins like HSP70, they protect the viral particles from degradation. So the viral particles safely sit in the insect gut crevices. And once after the insect visiting the new plant, the viral particles travels to the snout, and again, when the insect makes a puncture in the plant phloem cells, immediately virus enters inside the phloem cells. Like that, the insect vector Bemisia tabaci helps the transmission of the begomovirus from one infected plant to another healthy plant. Like that, the virus spreads from one plant to another plant. The next slide, I have shown the genome architecture of begomoviruses. DNA A, DNA B, and beta satellite of Begomoviruses. I have shown various types of RO marks showing different types of viral genes present inside the viral genome. The viral genes were encoded in the both directions, both the clockwise direction and anti-clockwise direction. And again, beta satellites also encode specific pathogenic proteins like beta C1. And in the case of alpha satellites, specific rep protein is there, replication associated protein, that is responsible for the replication of the associated viral genome. In this slide, I am proposing two different types of models for the possible movement of a viral DNA from the infected cell to new adjacent healthy cell. How two models are working? The first model is the couple skating model. In this model, the code protein transports the viral DNA from the nucleus to the cytoplasm. Once after reaching the cytoplasm, the code protein again joins with the movement protein of the particular virus. And again, this code protein, movement protein, viral DNA trio complex enters inside the plasmodesmatal openings. Here you are seeing the plasmodesmatal opening. And again, after reaching the plasmodesmatal channels, they reach another cell, because the plasmodesmatal channels are a kind of road. They connect one cell to another cell. Through this gap, the plant viruses escape from one cell to another cell. Begomoviruses also follow the same rule. The code protein, movement protein, and viral DNA complex enters through plasmodesmatal channel and reaches to the another healthy cell. Again, the viral pathogenesis starts. In another model, what happens in the case of relay race model? The code protein transports the viral DNA from the nucleus to the cytoplasm. So here, in the cytoplasm, the code protein DNA complex dissolves. The DNA is catched by the movement protein. The movement protein and the plant viral DNA reaches the plasmodesmatal channels and again traverse the gap and again reaches to the new healthy cell. There, this complex again dissociates and again viral DNA is surrendered to the new code protein, and again the code protein enters inside the cell nucleus and the pathogenesis starts. This is in the case of relay race model. Like that, various types of proteins, they perform multifunctional things inside the plant cell. Here, we have seen many types of aspects regarding the begomoviruses. We have seen begomoviral disease symptoms, infected crops, and how the virus get transmitted from one cell to another cell. We have discussed about the particle morphology, again genome characteristics, and how these viruses stay stably in the nucleus. Again, viral protein coding genes also I have explained. And again, viral replication in the phloem cells that we have discussed. So this much aspects we have discussed, and hope you understand better about the begomoviruses. In the next class, we will see about another virus. Thank you.